To cycle 2 of module 5 for investigating science. We're going to start with our do now for today, and I want you to look at these pictures and look at them and think which one of these best represents the work done by scientists and what makes you say that. Now, there may not be a right answer, but I want you to be able to defend what your choice is. Looking at our syllabus descriptors for today, we're starting to look at methodologies, so the process that scientists are using specific to their investigations that they're conducting. So we're again going to look at peptic ulcers, but we're also going to then start to look at the Earth's circumference, the Doppler effect, and Priestley's experiments with oxygen. And we're going to look at the methodologies that are possibly used in different scientific investigations and try and evaluate why we'd use those in different situations. And then lastly, we're going to look at what does it mean to have a peer-reviewed article. And again, we're evaluating our methodology, so we'll get to that verb later in our presentation. So scientific methodologies, we have 6 written down here. So obviously primary investigations, which can be done by individuals or groups. And an example here would be those conducted by Marshall and Warren with their peptic ulcers. We can do field work, so direct observations of interactions without actually changing any of the conditions. So letting, natural things happen and then taking those observations and analysing them. And that's something that Jane Goodall has done when she studies chimpanzees. We're looking at locating and using information sources, so examining the work of other scientists. These would also be known as secondhand investigations. And an example of this is the Human Genome Project. Surveys, collecting data from groups of people, modeling to be able to represent how things work, and simulation, so recreating parts of a phenomenon and then changing aspects. Example there we have climate modeling. If we change the different criteria there, we can see what would happen in the future and almost as a prediction here. So when we evaluate things, we're making a judgment based on the evidence that we have, and then being able to justify those, so supporting our argument with evidence. So when we're looking at whether methodologies are effective, I guess we're looking at whether they produce the intended result or whether they're ineffective, so not producing any significant or desired effects. And we can look at their evaluations, we can evaluate them in a positive or negative way. And we've got some words there that you may want to think about when you're making those evaluations. Then we move on to peer-reviewed literature. So peer review is a process where other scientists evaluate the quality of another scientist's work. To do this, they need to make sure that they're looking at all the different research and making sure that the article, if it was published, would add to what we already knew. Pretty much every scientific journal has the peer-reviewed system, and it has to be done blind, which means that the evaluators don't know the author. So it really is an impartial process. So we have the desk evaluation stage, the blind review, and then any revisions can be suggested. So, and a fun fact there, Nature is a science journal, and it usually only accepts 7% of submitted papers. So this peer review process is pretty brutal, but also quite rigorous to make sure that the journal is publishing documents that are high quality and high-end scientific methodologies and data and evidence. So we're going to be looking at some of the 4 scientists that we mentioned earlier. We're going to be looking at the methodologies that they undertook, and then class will evaluate whether they were appropriate for what they wanted to find out. So Marshall and Warren undertook primary investigation, so being able to find that link between the microorganism and peptic ulcers. So they did this by biopsying patients. What they actually did was human testing, so therefore part of their methodology was in fact unethical. And by taking the biopsies before and after treatment meant that they could compare those and look for the bacteria in each patient. Next, we have Arachnoids and the Earth's circumference. So he was using these techniques to try and work out the circumference of the Earth. So in a place called Syene, which is now called Aswan, the sun was directly overhead at noon in the summer solstice, so there was no shadows. At the same time, in Alexandria, he measured the shadows that were produced at the same time, which was about 7.2. So by using that calculation, he was able to also use the distance between the two cities and work out exactly the circumference of the Earth. We now know that's very close to the actual value. And his investigation was in fact practical. So he was able to actually conduct measurements, but also he was using secondary sources with the distances that he had there as well. And in fact, using that original calculation would have come from a secondary source. We then have Doppler and the Doppler effect. So the Doppler effect is where there's a change in the frequency of the wave in relation to an observer, which is moving it relative to the wave source. So he noticed that light spectra coming from stars was influenced by the movement of the star. If the star was moving away from the Earth, it appeared to be redshifted, so the light waves spread out, they decreased the frequency and increased the wavelength. And then if the star was moving towards the Earth, it became blue shifted, so the light becomes bunched, increased frequency, and shortened wavelength. So we see here that this would be creating our blue shift, and back here we're seeing our red shift. So again, taking those observations and looking at things in that natural environment. So he would have been undertaking, I guess, that field work, but on a much larger scale. And then lastly, we have Priestley and his experiments with oxygen. So Priestley was credited with the discovery of oxygen when he was working out how things burn. One of his experiments involved testing which gases could sustain life, and we have his bell jar experiments that we have here. So the same air that sustained the flame was also sustaining the mouse. So we can see that in B here, when the candle ran out of oxygen, the mouse died. And we can see here that the plant here actually survived in both of these parts. So the variation was when the mouse and the plant were placed in sunlight, the mouse was in fact safe. So in fact, the plant was refreshing the air. And again, we're coming back to this idea of the dephlogisticated air, which we looked at in our Year 11 course. And what in fact he discovered was oxygen, and then the fact that the plants were recycling that there to be able to sustain life, but also sustain the candle flame, allow it to keep burning with the oxygen. So this is a primary investigation, and whether this was effective or not, we will evaluate in class. So finally, making sure you update your inquiry question log using the descriptors at the start of the presentation, and completing the Google form to check your understanding against past paper questions.